Pleasure to introduce Dr. Hans Vink, who will lead the first training of these segments. Enjoy.
My name is Hans Vink, and today I'm going to present the first webinar in a series of 3 webinars on the microvasculature and glycocalyx. So today I will focus on the science that led us to work on glycocalyx and also the methods that we've developed to visualize glycocalyx and to measure its functional implications. So what happens with your microvascular system when glycocalyx breaks down, and how relevant is this for endothelial function, and how might this change and affect a patient's health? When we talk about the vascular system, we obviously have the large vessels as shown on the left. So 1% of our vascular system is the large pipes taking the blood from the heart to the extremities and to the organs, and then the veins carrying the blood back to the heart again. Well, if you zoom in with a microscope and look more closely, you'll see that most of your vessels are very tiny microscopic capillaries. And in these capillaries is where the action takes place. All the nutrients are transported through the walls of these capillaries to your tissue cells, and the oxygen is transported to the tissue cells, and the waste and the carbon monoxide is removed from your tissues into those capillaries and then flowing back through the veins, back to the heart and the lungs. Now, when you zoom in further in these smallest capillaries, you'll see that if you look at these, this is a single capillary showing individual red cells flowing through that capillary. And the topic of today is going to be glycocalyx. And glycocalyx is the compartment that in between the column of the red cells and the luminal surface are made up by the endothelial cells of these tiny capillaries. We worked on glycocalyx starting in the late 1980s, working on testing the hypothesis that indeed something was sitting in between this space. And I'll show you some of those early studies supporting the concept that a big glycocalyx occupies maybe up to 2/3 or 3/4 of the volume in the microvascular space under normal healthy conditions, and how that might change under different challenges. And then in the second and third webinar, I'll focus a little bit more on the GlycoCheck system, which is a medical device that we've developed to measure the health of your glycocalyx and microvasculature in patients. And the third lecture will be on endocalyx, which is a natural supplement that we've developed to repair a damaged glycocalyx and improve the health of your microvascular system, hopefully also keep your organs and overall health in a better shape. Now, when we zoom in a little bit into this space between the red cells and the endothelium, as shown with this image taken with an electron microscope. Again, you see here on the left a cross-section of a capillary, on the right-hand side a bigger magnification. So on top would be where the red cells are flowing. On the bottom here is the endothelial cell lining, which forms the inner surface of your microvessels and also the larger vessels. And then on the surface of that endothelium, in between the vessel wall and the compartment in the center of the vessel where the blood is flowing, you'll see these extensive 3-dimensional polysaccharide matrix, which is the glycocalyx. So those are polysaccharides, predominantly heparan sulfate and hyaluronic acid, produced by the endothelium and anchored into the membrane, then sticking out into the lumen. And these polysaccharides are highly negatively charged, and they'll bind plasma proteins, and then the plasma proteins will attract fluid to generate a hydrostatic pressure within that matrix, forming a protective gel protecting your vascular system. So in this lecture, I'm going to talk a little bit more about the history of the discovery of this glycocalyx. And then in the next 2 lectures, I will focus on the GlycoCheck system as shown on the left, which is a small handheld video microscope that we can use to measure live the way the red cells are flowing through your capillaries in the sublingual tissues in your mouth, and that calculates a score for glycocalyx and microvascular health. And then the third lecture will be about endocalyx, our natural supplement to repair and restore the health of your glycocalyx and microvascular system. Now, if I'm going to talk about glycocalyx and the studies that we did in the beginning, I have to mention this individual. This is a great guy called Brian Dooling, and he was working at the University of Virginia when I entered the field of studying microcirculation. So Brian already started working on observing microvascular health and red cell perfusion in the capillaries as in the early 1970s. And people started using big microscopes looking into capillaries. And he designed a few elegant and crucial experiments to demonstrate people that in addition to the red cells and the plasma flowing through the capillaries, it was highly likely that something else was sitting inside those capillaries, occupying a lot of volume and thereby modulating the number of red blood cells that are being present in those capillaries. And the reason that he started thinking about it were movies like these. So this is a live movie from a big microscope with a camera, looking in great detail on how blood is flowing through the smallest microvessels, the capillaries, in your tissues. And here this illustrates, if you look at those red blood cells flowing through a capillary blood vessel one by one, that if you take a snapshot of this picture, you would estimate that the red cells only occupy a small percentage of the total volume in these microvessels, and that most of these microvessels might be empty. So that triggered that discussion. So why are there only so few red blood cells present in these small capillaries? Another observation that he made together with his student Bruce Klitzman already in the 1970s, 1979 is when this paper was published. There was a paper published on the microvascular hematocrit, so that's the volume percentage of red blood cells in the capillaries. And not only that it was low under resting conditions, as shown on the left, only a few red cells present in the capillaries and only a few capillaries actually showing red blood cells, shown in the little movie before, but that also, if you would stimulate a muscle to contract or increase blood flow, metabolic products like adenosine, then instantly the red cell content in these capillaries could increase 3 up to 4-fold. But again, that triggered that discussion, what is responsible for the low and variable red cell content in these capillaries. This slide shows some of those original data that he published. So on the vertical axis is the capillary hematocrit, which is the volume fraction of red cells inside these capillaries. And on the top right, you see there's a dotted line where it says systemic hematocrit, 50%. So that's the volume fraction of red cells that you would expect to take a blood sample from a large vessel. Take a blood sample from a large vessel, put it in a centrifuge. You'll see that 50% of the volume of the blood is red blood cells, and the other 50% is a transparent liquid called plasma. Now, he was comparing that 50% with the estimates of capillary hematocrit that he got from observing the capillaries, by counting the number of red blood cells in these capillaries. And he did that under different conditions of external concentration of oxygen. So he measured that at resting conditions, on the low oxygen conditions, only 10% of the volume of the capillaries was occupied by red blood cells. And he also observed that if he made the muscle start contracting by electrical stimulation, that the red cell content could pretty much double in these capillaries. And in addition, he noticed that if oxygen concentrations would go up, blood flow would slow down, and also the red cell content would be even less than before. So drop down from a 10% to about 6 or 7%, also as shown in that little cartoon. And then again, also under low oxygen concentrations or high oxygen concentrations, when he started contracting the muscle, again, red cell concentration would increase 2 to 3-fold upon muscle contraction. While in addition to the effects of muscle contraction, he also noticed that he could increase the hematocrit further by adding adenosine, which is a metabolic vasodilator. So if the muscle starts working hard, metabolism increases, adenosine is produced, and that further increases blood flow. And in addition to increasing blood flow by dilating the larger arterioles and arteries supplying the muscle, supplying the blood to the capillaries, we added adenosine on top of the contractions. He could further increase capillary hematocrit up to 40%, getting very close to that 50% that you would expect based from blood samples from larger vessels. And that again is summarized in the schematic on the right-hand side. So on the vertical axis is the different levels of capillary hematocrit that he measured. And on top is the high levels of capillary hematocrit that he would measure during contraction together with adenosine. In the middle is the levels of hematocrit he would measure just by contracting the muscle. And on the bottom is the low levels of hematocrit that he would measure under resting conditions. And then again, the 3 different lines shown here are the predicted hematocrits in the capillaries based on the velocity of the red cells relative to the velocity of the plasma. Because if you start out with a 50% hematocrit in a vessel, similar as the systemic hematocrit, and then if you would increase the velocity of the red blood cells relative to plasma, that also would apparently dilute the blood. The red cells would just separate at a greater distance because the plasma is slower, and a normal estimated ratio of red cell velocity over plasma velocity would be about 1.5. Typically, the red cells flow in the center of the vessels, and the plasma is more sticking to the wall and flowing a little bit slower. So that would account that instead of 50%, you would measure about 40% in the microvasculature, similar values as he found during contraction and during adenosine, and that even if the red cells would speed up more relative to plasma velocity, so the ratio of red cell velocity over plasma velocity would increase from 1.5 to 2, that in theory could only account for a drop down to 30% of the capillary hematocrit relative to the systemic hematocrit in our larger vessels. So taking the hemodynamics into account could only explain this initial drop from 50 to 30%. It cannot explain the big discrepancy between the values that you would estimate at the lowest based on the hemodynamics of 30% and the very low hematocrits of 10% in the capillaries under resting condition. Additional studies needed to be designed to test whether on top of red cell velocity relative to plasma velocity, something else could be responsible for lowering red cell content. And one theory he came up with, that is, if you have a cross-section of a capillary on the top right here in the image, if you would hypothesize that near the endothelium, you would have a immobile plasma layer, so part of the fluid would not be moving and in some way stuck to the endothelium and just occupying space, that occupy a lot of the volume inside these capillaries and limit the volume available for red blood cells. So on the horizontal axis, it shows theoretical dimensions in microns of that immobilized plasma layer thickness. And then based on this theory that some of the volume would be occupied by an immobilized fluid compartment near the endothelium, he estimated that you needed a dimension of this immobilized plasma array of about 1.2, 1.4 microns to explain the low red cell contents for the different ranges of red cell over plasma velocity. But at this time was just an hypothesis, and nothing whatsoever was present in kinds of evidence that something like this might be true. Well, another explanation that he needed to test and disprove first was that the low red cell content was actually a dilution of the blood into the capillaries. So hypothetically, it would be possible that many of the red cells would be shunted by large vessels from an artery to a vein directly and not passing through the capillaries at all. So the blood would be really diluted with a low red cell concentration in the blood flowing through the capillaries. So to test whether that was also part of the low red cell contents, he designed this experiment, which he developed very, very small micropipettes, so hollow glass tubes, the very small tip at the end and a very sharp tip at the end of the size of an individual capillary. And he used these small micropipettes to puncture individual capillaries and to collect the blood that was flowing through the capillaries to test whether the red cell content in the blood flowing through the capillaries was similar or lower than the blood collected from large vessels. So the findings of those measurements are shown in the graphs on the top right. We punctured microvessels. We started out with smaller arterioles of about 100 microns in diameter. And from left to right, we moved to smaller vessels up to 10 microns or less, to true small capillaries. And also he measured going from the capillaries into the venular part, the venules of different diameters up to 100 microns. What this graph shows, if you measure the red cell concentration in the blood collected from these microvessels, as shown in the lower two graphs in this panel, so-called discharge hematocrit, so the hematocrit of blood discharge out of the capillaries, was still close to 50%. And if